today's class, we shall talk about the musculoskeletal system with a special reference to the human skeletal system here now. You know very well that muscles, the entire muscular tissue originates from mesoderm except a few muscles here now. The muscles like iris, ciliary body, iris and ciliary body, these two muscles originate from ectoderm. They are present within the eyeball here now, and the rest of the muscular system or the muscular tissue originates from mesoderm. Except these two muscles, rest of the muscles in human body originate from mesoderm here now. Now each muscle fiber, this is a muscle fiber here now. Each muscle fiber is covered by a connective tissue sheath like this. The connective tissue sheath is made up of collagen fibers, more number of collagen fibers and less number of elastin fibers. So the connective tissue sheath that covers each muscle fiber is called endomysium. Now many muscle fibers come together. Many muscle fibers like this come together to form a bundle. This bundle of muscle fibers is called fascicle. The bundle of muscle fibers is called fascicle. Now each fascicle is covered by a connective tissue sheath. That is, there is a connective tissue sheath that covers a bundle of muscle fibers, that is fascicle, which is called perimysium. And many fascicles, this is another fascicle here now. Many fascicles are held together by an outermost connective tissue sheath like this. Many fascicles are held together by an outermost connective tissue sheath called epimysium. So dear students, remember most important point here now, each muscle fiber is covered by endomysium. A bundle which is called fascicle is covered by perimysium. Many bundles are fastened together by an outermost connective tissue sheath called epimysium. Now this epimysium extends inwards in the form of perimysium and endomysium here now. Then it extends into the muscle from one side and comes out of the muscle from the other side like this. At one end it is attached to the bone. Okay, at the other end also you may find a bone here now. The bone which remains stationary is called point of origin. This is point of origin. The point of attachment of the muscle to a bone where the bone is stationary is called point of origin. And the point of attachment to a bone where the bone is movable is called point of insertion. Usually at the point of origin, the bone remains stationary. At point of insertion, bone moves from place to place. Like that, the entire muscle is attached to the bones now by a bundle of connective tissue fiber, that is more number of collagen fibers and less number of elastin fibers. This bundle can be called tendon. The bundle of connective tissue fibers can be called tendon. So in the human body, the muscles are connected to the endoskeleton by tendons. So at the point of origin, the bone more or less is stationary. At the point of insertion, the bone is movable. Okay, and in certain skeletal muscles are attached to the bones by sheet of connective tissue fiber. These are cord of connective tissue fibers. Certain muscle fibers are connected to the endoskeleton by sheet of muscle fibers, a sheet of connective tissue fibers called aponeurosis. So remember students, in case you get a question in the EAMCET or other exams aponeurosis is a sheet of connective tissue fibers by means of which a muscle is fastened to a bone, attached to a bone. So if it is in the form of a sheet, you call it aponeurosis. If it is in the form of a cord of connective tissue fibers, it is called tendon. And remember, EAMCET point of view, AFMC, JIPMER and other exam point of view, the study of muscle fibers is called myology. The study of muscle fibers is called myology or sarcology. Sarcology or myology deals with the study of muscle fibers. And the branch of science that deals with the movement of muscle fibers, the movement of bones, so on and so forth, that is kinematics part of the musculoskeletal system, is called kinesiology. This is another important point, students. Remember, EAMCET point of view, kinesiology. Okay, is a branch of science that deals with the movement of muscles and movement of bones, kinesiology. And study of muscles is called myology or sarcology. Same way you have got another branch which deals with the study of tendons and ligaments, which is called syndesmology. Syndesmology is a branch of science that deals with the study of tendons and ligaments. So remember, students, EAMCET, AFMC, JIPMAR, COMET, so on and so forth. The most important points kinesiology deals with the movement of muscles and bones. Sarcology or myology deals with the structure and their functioning of muscle fibers. Syndesmology deals with the tendons and ligaments. Now you have learnt about a fascicle, perimysium, endomysium and epimysium. Now we shall talk about the structure of a single muscle fiber. Now this muscle fiber here now, it is a modified myoblast. Okay, it arises from a mesenchymal cell called myoblast. This myoblast differentiates to form a muscle fiber. The cytoplasm of the muscle fiber, this is the cytoplasm content here now. Cytoplasm of the muscle fiber is called sarcoplasm. And within the sarcoplasm, there are several contractile filaments. Like these, several contractile filaments are present, arranged parallel to each other. Several contractile filaments are present like this, arranged parallel to each other. These contractile filaments, which are embedded in sarcoplasm, arranged parallel to each other, are called myofibrils. Myofibrils. Now within the sarcoplasm, there are mitochondria, several mitochondria are present like this. These are the mitochondria embedded within the sarcoplasm. The mitochondria of muscle fiber are called sarcosomes. The mitochondria are known as sarcosomes. Apart from this, there is endoplasmic reticulum. Like this, there is an endoplasmic reticulum. The endoplasmic reticulum of muscle fiber is called sarcoplasmic reticulum. Sarcoplasmic reticulum. The endoplasmic reticulum of muscle fiber is called sarcoplasmic reticulum. Then within the sarcoplasm here now, there are magnesium ions, Mg+2 ions are present. Then apart from this, a respiratory pigment called myoglobin is also present here now. In certain muscle fibers, a respiratory pigment called myoglobin is also present. The mitochondria are known as sarcosomes and endoplasmic reticulum is called sarcoplasmic reticulum. And dear students, remember, the skeletal muscle fiber is syncytial in nature. That is, it has got several nuclei arranged in a linear series below sarcolemma like this. If it is a mammalian muscle here now, in the mammalian muscle, several nuclei are found arranged in a linear series below sarcolemma. Now what is sarcolemma? The plasma membrane of the muscle fiber is called sarcolemma. The plasma membrane of the muscle fiber is called sarcolemma. Below sarcolemma, there are several nuclei arranged in a linear series if it is a skeletal muscle fiber of mammal, and if it is a skeletal muscle fiber of other vertebrates, the nuclei are scattered. Like that, you differentiate between skeletal muscle fiber of a mammal and that of other vertebrates on the basis of arrangement of the nuclei. Remember students, if it is a mammalian muscle, nuclei are found arranged in linear series below sarcolemma. If it is a muscle of other vertebrates, nuclei are found scattered in the sarcoplasm here now. Okay, and the only muscular tissue among three kinds of muscles here now, only the skeletal muscle is syncytial. The other two muscles, that is cardiac muscle and visceral muscle, they are uninucleated. These myofibrils here now, each myofibril bears alternately arranged dark bands and light band. There is a dark band here now. This is the dark band. All the dark bands are arranged at one place and all light bands are arranged at one place. Like this, these are the dark bands. These are the dark bands here now. All dark bands are at one place like this. All dark bands are at one place and all light bands are at one place. These are the dark bands. As there are dark bands here now, alternate arranged with light bands. Because of the arrangement of dark bands and light bands extending across the muscle fiber, the entire muscle fiber appears striated. That's why the skeletal muscle is also called striated muscle. Now these dark bands are known as A bands. The dark bands are known as A bands and light bands are known as I bands. Light bands are known as I bands. So this is the gross structure of a skeletal muscle fiber. Now we shall talk about the microscopic structure of a skeletal muscle fiber. So this is the microscopic structure of a skeletal muscle fiber. You have a dark band from here to here. This is the dark band, A band, and from here to here you have I band here. Now what is A band here now? A stands for anisotrophic band. Anisotrophic band, which is also called dark band. And I stands for isotrophic band or light band. Okay, this banding pattern is identified by viewing the muscle through polarized microscope here now. In polarized microscope, the dark band appears light and light band appears dark. In simple compound microscope, this particular A band appears dark and light band appears light in color. So on the basis of the polarized microscopic structure, the names are given here now. So A band stands for anisotrophic band or dark band. I band stands for isotrophic band or light band. Now if you look at the A band here now, is the entire A band dark? It is not dark. It has got a light zone in the middle. The middle light zone of A band is called H zone. Within the H, the thin feeble line is there passing through this. This thin feeble line that passes through this is called M line. So I repeat once again. The middle light zone of A band is called H. Within the H, the feeble line that traverses is called M line. Now what does H stand for? H stands for Henson's disk. H stands for Henson's disk. And M stands for mesoframe line. M stands for mesoframe line. Or it is also called Hayden-Hayne membrane. Hayden-Hayne membrane. mesophrene line or head and hair membrane. In the same way, within the eye band, that is isotrophic band or light band, a dark striated line is noticed. This dark striated line that passes through the eye band is called Z line. Is called Z line. Now Z stands for crosses membrane or Dobie line, Dwiston-Shaby line. Okay, Z stands for crosses membrane or Dobie line or Dwiston-Shaby line here. That is Z here now. And part of the myofibril that lies between two Z lines. Part of the myofibril that lies between two Z lines is called sarcomere. Part of the myofibril that lies between two successive Z lines is called sarcomere. Sarcomere is the structural unit functional unit of muscle fiber. So remember students, sarcomere is the functional unit of muscle fiber. Sarcocyte or myocyte is the structural unit of muscle fiber. So you must remember these 2 points here now. EAMCET, Comet K, AFMC, JIPMER and all the exam point of view. Remember, sarcomere is the functional unit of muscle fiber. Sarcocyte or myocyte is the structural unit of muscle fiber. Sarcocyte or myocyte is the structural unit of muscle fiber. And what is sarcomere? It lies between two Z lines. It includes a complete A band and two halves of I band here. Complete A band and two halves of I band. So with regard to microscopic structure of A band and I band, where A stands for anisotrophic band or dark band, I stands for isotrophic band or light band, H stands for Hensen's disk here now, M stands for mesophrene line or head and hair membrane, Z stands for crosses membrane or Dobie line or Dwiston-Shaby line here now. Okay, and remember students here now, part of the myofibril that lies between two Z lines is sarcomere. Sarcomere is the functional unit of muscle fiber and sarcocyte or myocyte is the structural unit of muscle fiber. Okay. Now we shall talk about the microscopic structure of A band and I band here. Now we shall talk about the microscopic structure of A band and I band here now. Now here I will draw an A band at the center and assume that from here to here you have an I band. I label this. This is I, this is also an I, and this is A. Now within the A band there are certain contractile filaments known as thick filaments or myosin filaments. Within the A band, the contractile filaments present are called thick filaments or myosin filaments. I shall label this. They are thick filaments or primary filaments or myosin filaments. You can make out in the diagram also students. Those thick filaments are confined only to the A band here now. They are about 100 angstrom units in thickness, 1.5 microns in length. 100 angstrom units in thickness and 1.5 microns long. They are confined only to the A band. And they are made up of a protein called myosin. They are made up of a protein called myosin. Made up of a protein called myosin here now. Now within the I band you have learnt already there is a striated line that passes through the I band, dark striated line that passes through the I band. This dark striated line that passes through the I band is called Z line. This is Z line, this is another Z line. Now this Z line in turn gives rise to thin filaments. These are the thin filaments that arise from Z line at one end, and at the other end they extend into the A band. Like this, you have thin filaments arising from the Z line and extending into the A band. Yes, these are all thin filaments. Now I shall label these, students. These are thin filaments, also called secondary filaments. Secondary filaments, also called actin filaments. These thin filaments are 50 angstrom units in thickness and 2 microns long. That's why they arise from Z line and extend even into the A band. And these thin filaments are secondary filaments. They are made up of 3 kinds of proteins. They are actin, troponin, and tropomyosin. What are they? Actin, troponin, and tropomyosin. They are the 3 proteins collectively form thin filament or actin filament or secondary filament. Now look at these diagrams, students. The thin filaments extend into the A band. In the A band, lateral sides both thick and thin are present. A band and the lateral sides both thick and thin are present. That's why A band, when you see under microscope, it appears dark on lateral sides and light in the middle. Because in the middle it has got only thick filaments, whereas lateral sides it has got both thick and thin filaments. We already learned that the middle light zone of A band, this light zone of A band is called H zone. The middle light zone of A band is called H zone. This is from here to here you have H zone. Now each thick filament is surrounded by 6 thin filaments. Remember, this is another important point, EAMCET point of view. Assume that this is the thick filament here now. Now this thick filament is surrounded by 6 thin filaments like this. Surrounded by 6 thin filaments. And the spatial arrangement of thick and thin is maintained by M-line protein. Maintained by M-line protein. There is a function of M-line. M-line that traverses through the H. M-line that traverses through the H. There is a thin M-line that traverses through the H. That is made up of M-line protein. M-line protein helps in maintaining spatial arrangement of thin filaments around the thick filament. Okay. So this is with regard to the molecular structure of thick and thin filaments. Now we shall talk about the structure of myosin filament and structure of actin filament here. Now we shall talk about the structure of myosin filament here now. Now each myosin filament, or which is also called thick filament here now, myosin filament, a thick filament, is made up of several meromyosin proteins. Made up of several meromyosin proteins. Meromyosin proteins. Made up of several meromyosin proteins. These meromyosins, around 200 to 300 meromyosins are there. 200 to 300 meromyosins are there in each thick filament here now. Okay. Now this meromyosin is of 2 types here now. We call it LMM and HMM here now. What does this LMM stand for? Light meromyosin. Light meromyosin and heavy meromyosin. Light meromyosin and heavy meromyosin here now. The light meromyosin forms the tail part of myosin filament here now. Each myosin filament, if you take, it has got a tail part. This is the tail. So the tail of myosin filament is made up of LMM here now. HMM forms the neck and the head. This is called neck and this part is called head. Okay, neck and head. This neck and head are made up of HMM, are put together called heavy meromyosin. So I shall repeat once again. LMM forms the tail part, whereas HMM forms the neck or arm. Neck is also called arm. Neck or arm and head part of the myosin here now. Now this head and neck, okay, are made up of heavy meromyosin, whereas the tail is made up of 2 spirally twisted light meromyosin proteins. These are the two spirally twisted, helically twisted light meromyosins now, and these are heavy meromyosins. Apart from this, around the neck, there are two kinds of proteins now. There are two kinds of proteins. You call them regulator protein and structural protein. So that is HMM is made up of altogether 4 kinds of proteins, 4 kinds of polypeptide chains, whereas LMM is made up of 2 kinds of polypeptide chains. So on the whole here now, how many are there? 6 polypeptide chains are there towards the head and neck part. 2 polypeptide chains are there towards the tail part here now. So the tail part is made up of 2 polypeptide chains, 2 polypeptide chains, then neck and head are made up of 1, 2, 3, 6 polypeptide chains. 6 polypeptide chains. Among 6 polypeptide chains now, the main proteins are in the form of neck and head part here now, and around the neck there are two more kinds of proteins now. You call them structural protein and regulator protein here now. Okay, they are called structural protein and regulator protein. So this is one structural protein, regulator, structural and regulator, which are spirally twisted around the neck part, and the neck and head are made up of another 2 kinds of polypeptide chains. So altogether 6 polypeptide chains are there around the neck and head here now, and only 2 polypeptide chains are there in the tail part here now. Now here this head parts of thick filament project out from the thick filament in the form of reversible structures, which are called cross bridges. That is, if you take a thick filament here now, this thick filament, all the tails are towards the center, towards the H zone, whereas the head parts emerge out like this from the thick filament in the form of reversible structures. All the head parts now emerge out from the thick filament in the form of reversible structures. All the tail parts are embedded within thick filament, LMM parts are embedded within thick filament, whereas the HMM part, that is heavy meromyosin part here now, along with its arm, projects out from thick filament in the form of reversible structures. These reversible structures are called cross bridges. The reversible structures are called cross bridges. So this is with regard to gross structure of thick filament here. I shall repeat once again. Thick filament is made up of, each thick filament is made up of several meromyosins, around 200 to 300 meromyosins are there. Meromyosin is of 2 types, light meromyosin and heavy meromyosin. Light meromyosin consists of 2 polypeptide chains which form the tail part here now. Heavy meromyosin is in the form of 6 polypeptide chains here now, where 2 of them are embedded in the form of neck and head. Another 2 of them are there around the neck in the form of spirally twisted proteins here now, where you call them regulator proteins and structural proteins here now. So altogether 6 proteins are embedded in the head part and neck part of the HMM, and 2 polypeptide chains are embedded in the tail part here now. Now these heads, tails are embedded within thick filament here now. All the tails of the thick filament are towards head zone, whereas the heads now along with the neck emerge out from thick filament in the form of reversible structures which are known as cross bridges. They look like tadpole like structures now. They emerge out from thick filament. Some of them are oriented towards Z line, and the remaining are oriented towards Z line of the other side here now. So the sarcomere, 2 Z lines are there. Some of them are oriented towards Z line of particular sarcomere here now, and the remaining are all oriented towards the Z line of the same sarcomere of the other side. Okay. Then what is the function of head here now? Each head has, number 1, active site for, it has got an active site for actin, that is, it can come in contact with the actin during contraction process. Number 2, it has got an active site for ATP, active site for ATP, that is, it can combine with ATP during contraction. Then it always carries an enzyme called ATPase. So these are the 3 functions attributed to myosin head here now. Number 1, it has got an active site for actin. Number 2, it has got an active site for ATP. Number 3, it carries an enzyme called ATPase, which helps in hydrolysis of ATP during muscle contraction. So it's all about the structure of thick filament here now, or myosin filament here. Now we shall talk about structure of a thin filament. Structure of a thin filament. As I made a mention earlier, thin filament is made up of three kinds of proteins now, namely actin, troponin and tropomyosin. What are they? Actin, troponin and tropomyosin here now. Now among these, the actin forms a major chunk of the thin filament here now. The actin is in the form of two polypeptide strands, spirally twisted polypeptide strands, which each polypeptide strand is called F-actin. F stands for fibrous actin. F stands for fibrous actin. Now, each fibrous actin is formed by polymerization of several G-actin molecules. So, several monomers like this, they are present here now. These monomer proteins which form the F-actin, these are the monomers here now. These monomers which form the F-actin are called G-actin molecules. They are called G-actin molecules. G stands for globular actin molecules. Okay. Several globular actin molecules come together to form F-actin or fibrous actin. Okay. Like this, there are two F-actin strands, where each F-actin strand is made up of several G-actins here now. Each G-actin has a binding site for myosin head here now. Then this F-actin at certain places is associated with a trimolecular protein like this. This trimolecular protein is called troponin. The entire trimolecular protein is called troponin. And what are the subunits of troponin? Troponin has, I shall give you the magnified structure here. Look at this, all of you. Dear students, assume that this is a G-actin. This is a G-actin. There are several G-actin molecules. Several G-actin molecules come together to form F-actin. Then as I made a mention earlier, each G-actin has got a binding site for myosin head. This is the binding site of myosin head. At this point, it can come in contact with the myosin head. Then this G-actin here now, okay, are in contact with a trimolecular protein, as I made a mention earlier, like this trimolecular protein. Trimolecular protein is there. This trimolecular protein is the troponin. Now we shall name them. This is TNI, TNC and TNT. They are the three proteins that constitute troponin complex. Troponin complex. Now what is TNI? Troponin inhibitor subunit. TNI stands for troponin inhibitor subunit. Inhibitor subunit. This particular thing binds conceals the binding site of G-actin and doesn't allow the G-actin to interact with the myosin when the muscle is at rest. That's why it is known as inhibitor site here now. I shall repeat once again. This TNI conceals the binding site of G-actin and doesn't allow the G-actin to combine with the myosin when the muscle is at rest. That's why it is referred to as inhibitor site. The next we have second one, TNC. TNC is troponin calcium binding subunit. Calcium binding subunit. That is, during contraction, calcium ions get attached to TNC to trigger the mechanism of contraction here now. The next we have another protein called TNT, that is troponin tropomyosin subunit. Troponin tropomyosin subunit. This troponin tropomyosin subunit here now partly comes in contact with TNC and partly comes in contact with another protein here now which lies in the groove of two F-actin strands, which is known as tropomyosin. That is, if you see this diagram here now, there is another protein which lies sandwiched between the grooves of F-actin. This particular thing can be called tropomyosin. Okay, it lies in the group of two F-actin strands. tropomyosin. It is made up of a single polypeptide chain, lies in the groove of 2 effectin strands. So like this, the troponin has 3 units. TNI, troponin inhibitor subunit, TNC, troponin calcium binding subunit, TNT, troponin tropomyosin binding subunit. And tropomyosin is a single polypeptide chain, lies in the groove of 2 effectin strands, and each effectin is called fibrous actin, formed by polymerization of several G-actin molecules. Each G-actin has got a binding site for calcium, each G-actin has got a binding site for myosin head, and it is covered by TNI when the muscle is at rest, with regard to the structure of thin filament. Now dear students, we shall talk about now the sliding filament theory that tells us about the mechanism of contraction in skeletal muscle fibers. The sliding filament theory, it was put forth by A.F. Huxley, H.E. Huxley, Niederjerk and Gene Hansen. So all of them independently have put forth this hypothesis, which is called sliding filament theory. A.F. Huxley, H.E. Huxley, Niederjerk and Gene Hansen. The biochemical events during muscle contraction, biochemical events that occur during muscle contraction were envisaged by Zen George. The biochemical events were envisaged by Zen George, whereas the theory was put forth by A.F. Huxley, H.E. Huxley, Niederjerk and Gene Hansen. Now according to them, the muscle fibers get excited by motor end plates. What are motor end plates? Remember students, all these terminology you should know, because they are all important for EAMCET, AFMC, JIPMER, COMET-K and AIMS, so on and so forth here. Now the muscle fibers get excited by motor end plates. Motor end plates. What is a motor end plate here now? A motor neuron ramifies to form several telidendrites. Now each telidendrite innervates a muscle fiber. Now all these muscle fibers put together may form a bundle. I learnt already, a bundle of muscle fibers is called fascicle. That is, each fascicle is under the control of one motor neuron, where each telidendrite penetrates into muscle fiber. It comes in contact with the sarcolemma of the muscle fiber. At the point of contact, it has a swollen structure. These are the terminal buttons. So the terminal button of telidendrite that forms synapse with the sarcolemma is called motor end plate. I shall repeat once again. The terminal button of a telidendrite that forms synapse with the sarcolemma of muscle fiber is called motor end plate. And the synapse that exists between telidendrite of a motor neuron and muscle fiber is called myoneural junction. The synapse that exists between the muscle fiber and nerve fiber or telidendrite and the muscle fiber can be put together called myoneural junction or sarcomuscular junction. All the muscle fibers of a fascicle are under the control of one motor neuron. When a stimulus is sent through this motor neuron, that stimulus makes all the muscle fibers of a particular fasciculus contract, and both of them together constitute motor unit. So remember students, the muscle fibers of a bundle which are innervated by telidendrites of a particular motor neuron, put together constitute motor unit. So the muscles will be under the control of motor units. Suppose this particular fascicle has 50 muscle fibers. This motor neuron which penetrates into the fasciculus has to have nearly 50 telodendrites, so that each telodendrite penetrates into a single muscle fiber, comes in contact with the sarcolemma of the muscle fiber in the form of motor end plate. And the synapse that exists between both of them can be called myoneural junction. Then entire unit, that is motor neuron along with the muscle fibers of a fasciculus put together called motor unit. Now, when the stimulus is made to pass to this telodendrite here now, it reaches the terminal button or the motor end plate and thereby depolarizes the motor end plate. Depolarizes, this is telodendrite, this is motor end plate, and here you have this is a muscle fiber, this is sarcolemma of muscle fiber. So here this is a synapse, motor end plate comes in contact with the sarcolemma to form myoneural junction. When the stimulus is made to pass to this, that is action potential is made to pass to this once the action potential depolarizes the presynaptic membrane of motor end plate, it activates the calcium ligand channels within the presynaptic membrane of motor end plate. Several calcium ligand channels are present. Calcium ligand channels which can be called calcium voltage gated channels. They are also called calcium voltage gated channels. Voltage gated channels. Now, when the action potential reaches, all these calcium voltage gated channels are activated. When the calcium voltage gated channels are activated, calcium ions present in the extracellular fluid, they diffuse into the motor end plate in large quantities. They diffuse into motor end plate in large quantities and thereby they facilitate the movement of synaptic vesicles towards the presynaptic membrane. Within the motor end plate, several synaptic vesicles are present storing the neurotransmitter. So when the calcium ions diffuse into the motor end plate, the synaptic vesicles move towards the presynaptic membrane. They come in contact with the presynaptic membrane and by exocytosis process, they discharge neurotransmitter into myoneural junction. So I shall repeat once again. When the action potential depolarizes the presynaptic membrane of motor end plate, calcium voltage gated channels of the presynaptic membrane are activated. Consequently, the calcium ions present in the extracellular fluid diffuse in large number into the motor end plate and facilitate movement of synaptic vesicles towards the presynaptic membrane. They come in contact with presynaptic membrane and by exocytosis process, they discharge neurotransmitter into myoneural junction. In most of the skeletal muscles, ACH, acetylcholine, acts as a neurotransmitter. This acetylcholine picks up impulse from the motor end plate and conveys the impulse to sarcolemma, that is depolarizes the sarcolemma here. Now from sarcolemma, the stimulus, the excitation, wave of excitation or the action potential is conveyed to myofibrils through triad system. Here students, you are coming across one more term here now. Remember, all these terms are important, their definitions are important as far as AFMC, JIPMAR, AIMS, then COMET-K, EAMCET are concerned here now. You have learned so far about motor unit, motor end plate, myoneural junction, okay. Then wave of the mechanism involved in conduction of impulse from motor end plate to sarcolemma. Once sarcolemma is depolarized, from the depolarized sarcolemma, the action potential is conveyed to myofibrils through triad system. Now what is triad system? In non-mammalian vertebrates, at Z line and in mammals at AI junction. I shall repeat students once again. In non-mammalian vertebrates at Z line, in mammals at AI junction, the sarcolemma invaginates into the sarcoplasm in the form of several transverse tubules. So sarcolemma invaginates into the sarcoplasm at Z-line in case of non-mammalian vertebrates. In case of mammals and human being, it invaginates into the sarcoplasm at AI junction in the form of several invaginations known as transverse tubules. Now these transverse tubules are in turn in contact with several cisternae. Suppose there is a transverse tubule like this. On either side of this, several cisternae are present here, like this. Several cisternae are present. So cisternae are present on either side, and the transverse tubules, all three of them constitute triad system. Dear students, remember the function of triad system here now. These invagination, the transverse tubules help in conduction of impulse, whereas the cisternae, all those small vesicles are cisternae, they store calcium ions, and on stimulation they discharge calcium ions into sarcoplasm to trigger the mechanism of contraction. So from the activated sarcolemma, depolarized sarcolemma, the stimulus or wave of excitation or action potential is conveyed to the myofibrils through triad system, which consists of invagination of the sarcolemma, that is transverse tubule, and the cisternae present on either side of the transverse tubule. And all the cisternae, always they will be filled with the calcium ions. On activation, on stimulation, they discharge calcium ions into sarcoplasm to trigger the mechanism of contraction here now.